Thank you very much for the introduction. I didn't do my disclosure. I work for CNL, which is Canadian Nuclear Laboratories. This is a national lab and at the same time operated by the company. So at CNL, we work towards availability, safety, and efficacy of targeted alpha therapy, and this is kind of a mission of our organization. I will start with a long history of what was before ACL and now CNL. We were at the front of innovation in radioisotope production and research, and launched medical isotope industry in early 50s to revolutionize how we treat right now cancer disease specifically. We pioneered production of cobalt-60 and molybdenum-99, which are critical until now still in medicine. And we were a major supplier of molybdenum-99 until 2018 when research reactor was shut down at Chalk River site. There were other isotopes which were also produced, xenon-133, yttrium-90, and so on. So with the closure of the nuclear reactor, there were a decision should be made what we do next, and CNL concentrated on 3 topics, which is to restore and protect the environment, develop clean energy, and also improve the health of Canadians. And this is what I will be talking about today. So some 10 years ago, there was this excitement about alpha therapeutics after a number of papers actually was published about success of different radiolabeled target compounds in first in human clinical trials. And on this picture, you can see that a patient was treated preliminary with lutetium-177 PSMA and didn't respond to a 2 round of treatments. And afterwards, after treatment with actinium PSMA compound, he responded and cleared from his lesions. And this excitement translated into the nuclear medicine community because now we can do much better radiopharmaceuticals, right? So at CNL specifically, we were interested in developing source of actinium-225, initially actually just for our own research. And this is some pictures from how it was actually done. So we have a number of, we have some waste on site which can be produced into something useful. So we used uranium-233 and separated thorium-229 out of it. It was used from old nuclear fuel. Proceeded with some chemistry, and after fine tuning and fine polishing, we were able to make a tiny generator, which is probably not as powerful as others, but still supplies a certain number of projects which are in-house, collaborative research agreements, and also we do some contract research for industry. So this is the scheme, the timeline. We harvest our actinium twice per month. One production is higher. This is where we actually milk our thorium generator, and we separate actinium, but also radium-225 is separated. And then we collect this radium, which then decays into actinium again and reprocess already radium to generate additional amount of actinium. So the production schedule is about once in 2 weeks. And if you ever received some of our actinium, it will be in a vial like this. And I don't know if you can see on the slides, but on the right photographs, there is a tiny blue spot at the bottom. This is actually Cherenkov radiation, which comes from the ionizing energy of actinium decay. So right now we make approximately 100 millicurie of actinium annually, and a majority of this is sent away for different projects, but we also do our own research. And as it was mentioned in the previous talk, capacity right now is not actually sufficient to feed preclinical and clinical trials. And current supply altogether taken from all 3 countries, from USA, Russia, Germany, and Canada, is only 2 curies per year. And it's not enough to bring into clinic even 1 drug candidate. So CNL saw an opportunity on this, and we also, as a research team, independent on business, were very interested in developing potential routes for this. So we engaged in the development of the actinium production. There are, well, I name only 4 production routes, and Paul, who will talk after me, will expand more about the production in general. Right now, there are 4 generators, as I mentioned, and there is a couple which are being in development, should be probably available soon, but still this is limited by the decay, physical decay of the isotope. Therefore, we cannot produce a lot. We want something more robust and to our schedule. There is a spallation reaction, which is also in development and in Canada here as well. It requires thorium-232, which is limited supply, of course, and there are not too many high energy accelerators installed. So again, we want to look at something, a broader picture where we can produce more or in addition to this. Cyclotrons is one of the obvious contenders. It requires radium-226, which is not easy to find, but also limited by target design technology. And at this point, it's very important to design targets which will sustain irradiation and the cooling. And electron accelerator, there are other groups who develop this technology. The same issues as, or the same challenges as with cyclotron production. So at CNL, we focus our research and development on cyclotron production. And again, I'm speaking here not on the behalf of business, but as a researcher who leads research teams. The idea at CNL was that we do have some legacy waste which contains radium, so we can probably separate this radium and transform it into useful targets. Then we can irradiate these targets in a cyclotron and generate material of suitable quality that can be sent away for development of radiopharmaceuticals. And cyclotron production is not new. It was studied previously, so it was more or less an easy choice, and the decision went into this direction. So in terms of radium material, we are using different kind of waste actually, which we can find, and that CNL owns a number of some amount of legacy waste. On the left side, you can see needles. This picture taken actually from the museum exposition. These are they made out of platinum or iridium, and they were used long time ago for brachytherapy with radium. So these needles can be processed, cut, and radium can be extracted. And at the bottom, you can see this different color peaks. So the red, you know, the red peaks show the spectrum. It's a gamma spectrum. It shows contaminated radium before it was purified. And after purification, you can essentially see only one peak in blue, which shows the pure radium. We also test it for chemical purity because we don't want to generate any additional radionuclides when we do irradiation of our targets. So we also wanted to measure cross sections, and this is to understand better what energy to use during the production. So we had to develop a tiny target containing about 20 microgram of radium, which was deposited on tiny film. And then if you look at the yellowish thing in the middle of the right top target, after 6 months just sitting on the table, this target became yellowish from exposure to radium itself. And at the bottom, you see a more reddish color. This is after irradiation in a cyclotron. So I should say this is really for research and development, and these are very tiny amounts which can help to measure cross sections. This is what we're actually very proud of. We call it Death Star. So to be able to measure cross sections accurately, we had to design, and this is our team of engineers and physicists, had to design a special equipment, a shielding, which will allow to measure everything very with precision to avoid recoils or other energy which surrounds the target during irradiation. So the result of this, we were able to identify, to measure cross sections and compare it with the calculations, which you can see in black line and then dotted line. And in the violet triangles, it's another publication. And for actinium-225, the blue squares, this is what we see as cross-section. And from our experience, it's that the peak energy is a little bit higher than the models predicted. We were able also to identify cross-sections for actinium-224, and for actinium-226, at least at this level of production, it was below the detection limit. So we could identify only the upper limit. Why actinium-224 and 226 are relevant? Because they are contaminants during the production, and they can, because they are shorter half-life, they can actually contribute to excessive dose, or just to contribute to a dose. I would not say excessive, but contribute to a dose of a patient. At this stage, we work on development, process development for use in a hot cell. You should understand that radium itself, radium-226, is a radioactive material. So comparing to PET production, where we can have a cold material, fill our target, and then do irradiations, here even the target preparation have to be done in a hot cell. And during the decay, there is also radon, which is generated, and it's volatile compound which can contaminate surrounding hot cell walls or whatever equipment is sitting in this hot cell. So we develop procedures and special equipment that will go inside the hot cell to facilitate the work of the operators. And this is a very recent news. Just 2 days ago, ITM and CNL announced the launch of a new joint venture, Actinier, which will be now taking care of the actinium production at a larger scale. So it will go from the hands of CNL to Actinier. And of course, research and development team always ready to support with our new developments. Now I will change a little bit to talk about the quality control. So you saw from previous presentation, and the decay chain is very complex. And when we submit our application, for example, to regulatory agencies for a new drug, we need to identify clearly what is the structure of the compound. And with actinium is not totally possible, and also very difficult to measure accurately how much actinium and how much daughter is inside. And this letter to editor was published a couple of years ago, actually no, last year. Specifies that accurate measurements of radiochemical purity for actinium radiopharmaceuticals still constitutes an unsolved problem in radiopharmacy. And this is very important to test it adequately, because alpha emitters have extreme toxicity. A little bit of physics, I guess, and this slide is inspired by one of my colleagues in Sloan Kettering, David Bauer, who was giving a talk on chemistry earlier at the CNM this year. So essentially, just with doing normal calculations using decay equations, we at the equilibrium we can establish approximately what will be the purity of our actinium. But all these francium-221 and astatine and all other daughters, they will probably, again, as mentioned during earlier talk, leave the chelator and wander around somewhere in the body. Right now, also, there is a lot of effort actually to test these compounds at the end, and we use very sophisticated instruments, gamma spectrometry, alpha spectrometry. Alpha spectrometry is very complicated even just to prepare a sample, or ICP-MS. And it's clear that not every nuclear medicine department or even production facility have this type of instruments or have qualifications or special expertise or people to do this. Therefore, first of all, decentralized production might make sense, but also we are trying to address it by developing more simplified quality control methods. And CNL, together with Triumph, actually, is part of International Atomic Energy Cooperation research program or project where we try to develop simplified quality control methods, standardized measurement techniques, and also develop release specifications which allow, independently of the production route of actinium, be able to be used for radio labeling and in clinics in the future. One other thing what we are aiming at CNL is also to develop chemistry and formulations. So essentially, at some point, we have something like kit-like radiolabeling procedures, and you may know about it from if you have experience with gallium and gallium generators when you receive cold kit and when you receive the isotope. So it would be, I guess, ideal situation in nuclear medicine department. We also started to look into calibration factors for different dose calibrators and certify them against actinium that we produce just to find the right calibration factor, because to be able to measure it in a nuclear medicine department, you need to enter this right button, right? And usually you even don't know the calibration factor. It just tells you, okay, this is technetium or this is lutetium. So we are trying to find out what is the calibration factor. So eventually this can be easy as a technetium measurement. Is specific activities important? And this question, of course, depends on the target which we're trying to target in the body, or biological target. This is comparison of compilation of different literature data, and in blue, this is the results we do consistently when you do production. We actually test for radiolabeling with dot calculator. So these are results, and you may see that if we really want to have like kit-like procedure, we probably not at the highest specific activity. If we add purification step, we can go a little bit over. But as experience with gallium and lutetium shows, for majority of targets, we even don't need high specific activity. So it should not stop us from developing radiopharmaceuticals. And I would like to say a few words about radiobiology and dosimetry. This is also what we do at CNL. We have a long-standing expertise in radiobiology and dosimetry coming from ages ago. And it's now acknowledged that the theory of, just double breaks of DNA, this is how alpha therapeutics works. It's not the complete picture. And it's also acknowledged right now that the radiobiology of gamma and beta emitters is not the same as high lead alpha particles. Therefore, there is a lot of research which still needs to be done on preclinical animal models to actually evaluate microdistribution effects and relative biological effectiveness and dose rate effects. There was a mention about bystander effect, and there is also systemic effects which are regulated by immune system. So we have several research projects which try to address these questions, both in vitro research and animal studies. And to add a little bit in terms to answer the question, I guess I asked at the beginning if it's the game changer, and yes, it is. But at the same time, I would like to show you this slide, and this is the project which was not related to development of antibody for clinical use. Essentially, there are already antibodies which are in clinical trials, but the idea of the project was totally different, to evaluate the effect of actinium on healthy stem cells to see if they will survive. And the different 6 cell lines were tested. On the top, HCC, it's stem cells. Jurkat is a negative control which doesn't express CD33. All other 5 express CD33. But if you look at the middle, at the concentration of 0.25, there are some cells which are sensitive to action of actinium-labeled antibody, and there are 3 which are not. And 1 is control, so this is what we expect. But other 2 cell lines actually something unexpected, and it means that there is something else, probably genetic background of each of these cell lines, which brings this resistance. So we are trying also to elucidate the mechanisms of why it happens, and this brings us to the discussion about combination therapies. Probably non-single radioisotope or non-single mode of treatment will cure the cancer. So we need to look at different combinations and what is possible. And this is something which also excites us a lot. And I think we will hear more about combination treatments tomorrow in the morning. There is a presentation. I guess this is close to the conclusion. Just to mention that we use our waste to make a tiny, again, research scale lead generator. And our initial attempts were successful. So right now our process is about 83%. And you can see the example of labeling with DOTA. DOTA chelator, this is a peak which shows that we have quite quantitative labeling, and the gamma spectrometry showing at the bottom the lead that was eluted from a generator. Again, underlining just for own research and maybe eventually for collaborative research, but this is not a business project or something that we plan to commercialize at this time. And a road ahead, I think CNL aligned very well with what community needs and what the, in general community where it goes. For us, reliable production of radionuclides, alpha therapeutics in sufficient quantity and quality is important, and we as researchers try to contribute to this. We try to develop detection and counting techniques and also standardization, also in terms of phantoms for imaging, for example. Research to understand the mechanisms of action to improve efficacy, and here are also combination goals and immune system effects. And this is basic research, but it can contribute to better, to more efficacious drugs. We are interested in developing imaging counterparts as well, or at least investigate if they behave similarly in the body, because there is a lot of interest in trying to find an isotope which will be ideal for this, and until now there is no consensus. And also radiobiology and microdosimetry. This is a totally rejuvenated field right now after success and interest in alpha therapeutics, and CNL has a great expertise in this, and we are hoping to contribute in this as well. So this is our collaboration and partners. Our research is mainly sponsored by EACL Federal Nuclear Science and Technology Program. We also have our internal CNL's lab-directed research funding, and we collaborate and partner with different institutions in Canada, and hopefully soon, well, there is already one internationally, but hopefully soon internationally. And I would like to thank my great team who actually does all this work. They're all young people and eager and very excited about our projects, and I would like to thank you for your attention, and I will be ready to answer your questions, I guess, at the end of the session. Right. Thank you very much.